Hey, so today we're going to talk about mutations. Before we do that, we're going to do a quick review of transcription and translation. You're going to need a piece of paper, something to write with, and a codon chart for this lecture. So if you don't have those out already, I'd like you to put me on pause and go grab them. So our first task is going to be to transcribe and translate this DNA to messenger RNA and then messenger RNA to amino acid sequence. Again, pause me, see if you can answer those questions. Go to messenger RNA and then use your codon chart to figure out the amino acids. And then come back and you can check your answers. For any questions like this, it's really best for you to do it yourself first, make sure that you know how to do it, and then check your answers. If you're just copying what I write down, you're not really checking to make sure you understand it. It's always easier when somebody else does it. So, do your answers match up with this? So remember that G always base pairs with C and C with G, but if we're dealing with RNA, instead of a T for thymine, we would have a U for uracil. So the A goes to U. T still base pairs with A in RNA from DNA to RNA. And then for your codon chart, if we're looking at so we have G, C, U. If you check here, you're going to look for G. You're going to look for G, C, and then U. So it's going to be ALA, and then ACG, find A, and then C, and where that meets, and then G, you come over, you have THR, and then UAG, you get a stop, so you stop. All right, so again, we went from DNA to messenger RNA to amino acid. If you struggled with this step, then I would encourage you to go back and review protein synthesis and base pairing rules before you finish the rest of this lecture. All right, mutations. So mutations are just changes in DNA, and they're going to affect the traits that we see. So remember, if DNA codes for messenger RNA, and messenger RNA is used to code for a protein or the order of amino acids in a protein, and those proteins results in traits, then if you change the DNA, you are going to change the messenger RNA, and you may change the protein. And if you change that protein, it may change the trait that you see. And so understanding mutations is important because then it allows us to understand how a trait got changed. So again, DNA to messenger RNA to protein, protein to trait. So a mutation occurs when there's a change in DNA, but what causes those mutations can vary. Sometimes it's just a mistake when DNA is getting copied that doesn't get corrected. Sometimes it's an error during the formation of gametes, sperm and egg cells, so that's a process called meiosis. And we'll get more into that later on. There's also environmental factors or things called mutagens that can cause changes to DNA. So when there's damage to DNA, when the enzymes go to repair it, sometimes they make mistakes. So things that tend to cause damage either physically or chemically to DNA are what we call mutagens because they increase the likelihood of mutation. So those can be things like chemicals, then a lot of those are found in cigarette smoke, ultraviolet radiation, X-rays, even some viruses have been known to increase the likelihood of mutation in the DNA. Now, usually when we think about mutations, we think about them being harmful, or we think we're going to be a superhero. Mutations happen all the time. Most of them get fixed, and then a lot of them are what we call neutral mutations. They don't actually change the protein that results, and so they don't really have an effect one way or the other. Again, many get repaired, but some can be harmful, and they can lead to disorders or cancers when the cells start growing out of control. Other mutations can actually be helpful. All of the variation that you see around you, whether it be in eye color or blood type, those are all the results of a mutation. So let's talk about the different types of mutations. And today we're focusing on gene mutations. So those are at the DNA level, the A, T's, G's, and C's. In a later lecture, we'll talk about something called chromosome mutations, and that's kind of like a larger scale. Again, the ones we're going to focus on are a substitution, addition, or deletion. And that's just what it sounds like. A substitution, you substitute one nucleotide for another nucleotide. An addition or insertion, you're going to add one. And a deletion, you're going to take one away. So again, a gene mutation is a change in the nucleotide sequence of a gene. It can involve a single nucleotide or multiple nucleotides. When we talk about a single nucleotide, we call that a point mutation. And again, the three that we normally look at are substitution, insertion, and deletion. Substitution, you substitute or exchange one for the other, so you put an A instead of a G or a C. Insertion, you add an extra nucleotide, or a deletion, you delete or take one away. Insertions and deletions cause what we call a frame shift. So they shift the reading frame. Since we read DNA and messenger RNA in 3 base words, if you shift or the frame of reading, then you're going to have an impact further down. So a point mutation is just a change in a single nucleotide. And even a small change, like a substitution, can have a huge impact. So here's an example called sickle cell disease. So in sickle cell disease, you have an A changing to a T. And so what happens is you end up coding for a valine instead of a glutamine. And this is really important because it's in the hemoglobin gene. And so what occurs is red blood cells are going to, instead of having this nice circular shape, have this sickled or crescent shape. And as you can imagine, these are floating around in your body. They get caught on things. And so there's a lot of complications that can occur. They're also not very good at carrying oxygen if they have this mutation. So this is going to be one of those that's just a substitution. You switch one base for another, and you end up with a huge impact on the individual. So that was substitution. Now let's look at insertion and deletion, these frameshift mutations. So here you can see substitution, you just change one amino acid. Frameshift mutations, you're changing that amino acid and everyone after it. So again, by inserting or deleting one or more nucleotides, you shift your reading frame, and those tend to have a greater impact on the overall protein. So let's look at it in just a normal sentence. So the fat cat ate the wee rat. Now if we add an A, we get the fat aka tat eth u era t. So by adding just one letter, we've shifted, and every word after that is impacted. Same if you subtract one. All right, so now let's practice with these. So this is the example we used at the beginning. So you should already have this written down. We're going to make some changes to this and see what happens. So here's our normal DNA. Here's our mutated DNA. See if you can answer these questions. So again, you're going to want to pause me. All right. So this is what we call a substitution. Notice this T is substituted, it's switched. So you go from an A to a T, which changes the messenger RNA, which changes the amino acid sequence. So that stop is now a less. Let's try with this one. Again, pause me, answer it, and then come on back. All right, so this is what we call an insertion. You can see that this A was added. Sometimes these are a little trickier to figure out. And that shifted my reading frame. So I'm going to change this codon, but I'm also going to change the codon after that. And you'll notice that that changes not only the last one, but the one before it. All right, how about this one? This is an example of a deletion. So we removed that G. And here, we've also changed that amino acid, and you can imagine if it was a longer chain, all of the ones after it. So see if you can define and give an example of each of the following: a substitution, deletion, insertion, and frameshift. Then see if you can answer these questions. Are mutations bad? And what causes mutations? Then, if you want to challenge yourself, there's something called a silent mutation. A silent mutation is when you have a change, but it does not affect the amino acid that you create. See if you can find an example of that, a change that you could make to a DNA sequence that would not affect the final amino acid. Remember, if you have any questions, you can always ask in class.